Hi, friends of the sun. Welcome to our weekly reading group happening every Monday. It's just on Zoom. You can join it yourself. We have an author of some interesting paper that I was curious about presenting their paper, discussing their paper with us and me. And yeah, today we're talking with Ali Saberi about RNA foundation models and why we now also need the RNA foundation models, apparently next to DNA foundation models. So let's go. Aha. Hello, hello. I think you're muted and I had my audio off. So now I should be able to hear you.
Hello there.
Hi, Ali. Very nice. Very nice. Thanks for
Nice to
taking
see
the
you.
time.
Yep.
So
How do you
yeah,
pronounce your
today
name properly? Hannes. Your name is Hannes.
that's pretty good. Hannes. Yeah.
It's
And
nice. See you,
uh,
Hannes.
I hope I can at least get three letters right, Ali.
Yep.
Okay. And Ezra is also here. Yeah, so I'll have to leave early today, like 15 minutes early, because of an RQE examination or like some PhD stuff. And then Ezra will be taking over. Well, Ezra, do you actually have time? Like in 45 minutes or so?
I have to run around then as well, actually, around
Yeah.
12:40, so a little bit before then, unfortunately.
Well, that's fine as well. Then let us just be efficient today and have it. We'll just chat 45 minutes. And yeah, I guess we're kind of well, I don't want to speak for everyone, but most of us, at least usually, we're more machine learning people, I would say. And like, so we don't really care so much about, okay, you now have your transformer here, and it's big and it's hyena and or whatever. And we all know how hyena works. We know our FFT mixing stuffs and so on. Um, but what we don't know and what we really hope to learn from you is like, why the hell do we now need an RNA foundation model if, I don't know, we already have EVO or so, which is a DNA foundation model? And how do we even, like, how is the DNA, RNA data even different from the DNA data? I mean, we know what are introns and exons and so on, but um, yeah, but how do we, if we do our sequencing data and you have your, I don't know, you said say some PacBio sequencing is, I think, what you use. And how do you even know where the introns begin and where they end? And how do you know like, where the whole, what the whole part of the, yeah, questions like this. We'll get to it later. But do you have slides today that you want to use, or do you want to screen share the paper?
Yeah, I actually like present this project several places with biological background and computational background. I merge
Uh,
all these slides to be comprehensive, so some of the information might be redundant, but I will skip through it. And I
okay.
guess well, I will answer these type of questions, but feel free to interrupt me, ask question to make sure that you get what you need.
Then how tolerant are you to me, like being like, no, 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 let's skip this slide as well. We don't care about that.
Yeah, of
Um,
course. Let's do that.
okay.
Let's be efficient.
Alrighties, but then yeah, let's get into it.
Maybe.
The thing is already recording, and we would put this up on YouTube as well, the discussion afterward. If you have something against it, let us know. Otherwise, this thing is going up, and you can just let me know via email as well. And then yeah, just let's start. And please also have a quick introduction of yourself so we can, yeah, all admire you the more.
Okay, thank you very much. So, hi everyone, nice to meet you all, and thank you for inviting me here. More than happy to present my main PhD research. My name is Ali Saberi. I'm currently a PhD student at McGill doing computational biology. I came from Iran, Sharif University, studying AI to here, and I'm collaborating with ARC Institute. So it's a big project, collaborating between McGill and ARC, and I'm working on this since 3 years ago. and we have some cool results I want to share with you, and hopefully I will answer the questions that you have in mind. So let me start with it. So the title is Long Context RNA Foundation Model for Predicting Transcriptome Architecture. Pretty fancy. So let me make this smaller. Okay, some basic biology. We all know about central dogma of biology. We have DNA that is transcribed into RNA. We have some type of RNAs called messenger RNAs or mRNAs that are translated into proteins. And we have co or post-transcriptional processes. And okay, go on.
So, right away, mRNA is messenger RNA, right? But now I think today the Nobel Prize for Medicine also was awarded to people discovering microRNA.
Yes,
Um,
yes.
what's
I remember.
microRNA?
So your question is what is microRNA?
Yeah, basically.
Okay, so the thing is, like this terminology in biology, it's somehow like the first time that they discovered that it was smaller than the previous RNAs. So like the average length of microRNAs are 20 nucleotides, something like that. So they call it microRNA. So they are RNAs, which means that they are transcribed from DNA, and these are single-stranded ribonucleotide acids. And the thing is, we have long non-coding RNAs, which is RNAs that are super long, and during some like lots of processes, these RNAs are going to be splitted into smaller parts. And recently, they found that these smaller parts actually have lots of functions. And they will, for example, one of the functions that I remember I read long time ago is that these small RNAs would go and bind to some particular mRNAs and make them like avoid them from being translated into proteins. Functions
Mm,
like this.
okay.
So yeah, but okay, we have something in chat. Hopefully, mRNA code for the linear amino acid sequence for the protein, mRNA code for post-translation.
Yeah.
It's like this is good. This is a good description, of course, but it's like when you look at the microRNAs like functions and usage, of course, one of them is post-translational regulations, as I mentioned. But I guess every day they find new functions for microRNAs.
Yeah. Okay,
Oh,
then let's continue. Sorry for that
okay. Of course,
slide.
of course. Like I try to share all of my biological knowledge here, but my background is also AI. So I have like among engineers, I have good amount of biological knowledge, but among biologists, I'm just a noob. So let me continue. And so the RNA splicing that I talked about, here I added this slide to make sure that we all know the terminology here. We have DNA double-stranded exons and introns, and then transcribed into single-stranded RNAs. And then we have, after lots of processing, we have like the translation that goes to protein. And one of the processes that happens co or post-transcriptionally is RNA splicing, one of the most important ones, and in which the introns are removed. And let me put in a laser. It's cooler. Yes, that's it. So introns are removed and exons are ligated to each other. So from pre-mRNA, we would have mature mRNA that then would be translated into protein, of course, after some other processes. And we have something called alternative splicing, in which, again, the introns are removed, but different combinations of exons can be ligated into the final product. So we would have different spliced mature mRNAs, and therefore we would have different proteins. And just to make sure we all know about these regions that are removed from pre-mRNA are called introns. We have these that are ligated, spliced in. We have exons, and these boundaries we call it the splice sites, which are really important in. Go on, Hannes.
Can it sometimes happen that we have overlapping genes and then we have like an exon of another gene inside of the intron
Yes,
of
yes, of
this
course,
gene?
of course. Yeah, like yes, based on my basic understanding, everything happens like it's possible, and we have overlapping genes, but like we can study them individually. So I will explain in the next slides, but we can say that, okay, let's consider that this is the gene, this is the start of the gene, this is the end of the gene, and these are the exons and introns. Of course, it could be overlap with other genes, and like some intron could have, for example, this exon that I indicated here, when it's not included inside the final product, we can see that, okay, this is an egg, this would be an intron for some isoform.
Okay.
I
Okay.
will
Next word,
next.
isoform. What's an isoform?
Okay, yeah, I will explain it. I'll explain. Yeah, and okay, so this is the explanation. So we have this gene. Consider that. Let's consider this like a simple scenario that there is no overlapping gene. We have this DNA, and some part of the DNA we have this gene, and as I explained, we have this splicing process that the introns are removed and some combinations of exons are selected and ligated inside the final product. Depending on this selection and this splicing product, we would have, okay, nice. Yeah, we would have this different isoform. We call this transcript isoforms because all of them are from the same gene, but the combinations of exons are different. So this gene could have this, for example, this is the reference isoform, which means that inside the human cells for this particular gene, we have seen these different combinations. So we call these are different transcript isoforms of this gene. Okay.
So isoforms are the different proteins that can arise from one gene.
Exactly, exactly, exactly.
Okay.
And so, and we have for this transcript, like we call this alternative splicing, and we also have differential alternative splicing, which in different tissues and cell lines and cell states, we would have different ratios of these transcripts to be produced. For example, here you can see this is an example, like in brain for this particular gene, which I guess is in BNL1, something like that. These two isoforms are dominant in terms of expression, and 80% of the products are from this particular type of isoform. But in liver, it could be different. So you can see how like we have nearly like 20,000 protein coding genes in human, but like these, our body, our cells are producing millions of different proteins from 20,000 genes, and this is the way that it works. We have different isoforms, so from one template of gene we could produce, you can imagine how different the outcome protein from this isoform would be, and with different ratios. Okay, and one part of biology that is really important, and that I will come back to this, how this happen, like how we would have alternative splicing and differential alternative splicing. It's all go back to the DNA and RNA, to the sequence. We have RNA bonding proteins and DNA bonding proteins. Like, for example, for the splicing, we call this a spliceosome. Like 150 proteins come together, constitute a huge complex to do the job, to remove the introns and attach the exons. And all of this RNA binding protein recognize motifs, the sequence features on the DNA, on the RNA, to understand, okay, where is the start side, where is the end side, and the availability of this RNA binding proteins in different tissues will end up having different isoforms in different tissues. So one particular, for example, in order to recognize this splice site for this particular exon, one particular RNA binding protein is needed. And for example, let's say in some tissue, this RNA binding protein is not available, so we wouldn't have this exon in any of the outcome proteins, outcome isoforms, eventually the outcome proteins. So I'm trying to, yeah, go on, Hannes.
For the spliceosome, you write it's a large protein complex, and you said 150 proteins. Should I actually imagine just a ball or like a single structure of 150 proteins doing the job, or is it more like we have maybe 20 complexes out of these 150 proteins that then?
The second one. The second one.
Okay.
Sorry if I said it wrong. Like I remember when I started this project, I read a Nature Molecular Biology review paper of splicing. I can find it and share it in this presentation, and it's like detailed biology and talking about every protein and every binding site and how they are like coming together. But if I remember it well, it's not like one huge complex. It's like, as you said, different parts come together.
Good, good.
Okay. Okay, I have this great green tea. Okay, so RNA splicing is super cool, super complex, but why it's really critical and essential and important, you may like have some clue based on this slides that I described, but like just one example here, we call this a cassette exon situation where we have three exons and the middle exon could be included in the final product or could be removed from the final product. We call this cassette exon included and cassette exon excluded. So this is one particular, okay, we have a comment here. Think of it as a scrambled letters exon that can spill.
I think it's not a question. Like if we have a
Okay,
question, then
okay,
I'll point it out in the
okay,
chat.
okay. So just a comment for other people to read. Thank you, Michelle. And so okay, so we have this cassette exon, and that based on all of the things that I mentioned, the product would be different. And as an example here, there's also another Nature oncology review paper that talks about splicing in cancer, which is pretty cool. Again, talking about how important splicing is in developing cancer. And here's an example in this MCL1 gene. You can see that these are the number of exons in this gene. And whether the middle exon, the cassette exon, is included or excluded, the final protein would have contrary functions. And it's super important for if some particular cancer wants to develop, this gene needs to have like more of this protein, the anti-apoptotic one. So you can see that like this is a really basic example to show you that how dysregulation or changes in the outcomes of splicing would cause a normal cell to eventually become a cancer cell. So this process is really important in order for the cell to like behave normally and act normally and healthy. So now this is how like starting of my project. So like the previous project, the previous slide, I got it from another presentation, some kind of like background description, and now starting the project. Transcriptome architecture, like this is the definition that we use. So based on all of the like description that I provide in the previous slide, the cell identity, the identity of the cell, which means that like how the cell is behaving, how the cell behaves in different situation is mainly determined by the transcriptome architecture. And what is transcriptome architecture is the molecular structure and the abundances of RNA isoforms. So as I explained, we have DNAs, double-stranded with these exons and introns, and then we have transcription and RNA. So this RNA would have a molecular structure. You can see that the exons, introns, and we would have alternative splicing, like different molecular structures are chosen and different abundances of them. You can imagine that, okay, I don't know why this happened. Okay, and based on this different abundances of this RNAs, mRNAs, mature mRNAs, we would have different abundances of proteins. And these proteins eventually, and their abundances, eventually determines how the cell behaves in different situations, let's say the cell identity. So you can see, we call this transcriptome architecture, the molecular structures, the combinations of exons and introns, and their abundances, call this transcriptome architecture, and we want to model this. And as I explained before, go on, Hannes.
You want to model this, but doesn't this now you said this is what defines what cell we are in, but doesn't what cell we are in also define what gets transcribed and what's like also determines all these transcript abundances?
Okay, can you ask me again? I'm not sure I
Like
understand.
the transcriptome architecture, it is what are the isoforms, like what are the isoforms that are built and
Okay,
how many of each isoform.
okay, yeah, yeah, exactly,
And
exactly.
now this determines the characteristic of the cell. This
Mhm,
determines the cell type, basically.
exactly.
But then what determines which isoforms are transcribed and which genes even are transcribed? This is also determined by the cell that we are in. and the environment that we are in and the
Exactly.
transcription factors and so on. So this is like sort of circular, no?
Yeah, definitely. So you're talking about, like, you're saying that I'm assuming that something is transcribed. So transcription happens, and then we have post-transcription or post-transcriptional things that during the splicing, which of these combination, which of these match pre-mRNA is going to be spliced
Mm,
and have different abundances, right?
I think I have my thing now figured out, right?
Okay.
I was like, the information isn't contained in your DNA because in every cell you have the same DNA. But maybe
Yeah,
you're
yeah.
saying the
No, no, I
information
think
is
that,
contained
go on, go on.
in is contained in the RNA after
Okay.
it's has been transcribed. And like if you are in one cell type, then only like this one gene gets transcribed and the other gene doesn't get transcribed. And now because maybe a transcription factor is missing or so, and now but we now assume that all the transcription happened. And now with your model, you only take RNA as input, and then from the RNA you want to predict what are the abundances of the isoforms and what are the isoforms.
Exactly, exactly, exactly. Completely, you're completely, like, one of the extension of this project that we have in mind. We're going to, like, publish the final version by October 21st, and then one of the extensions is to just extend it from RNA to DNA also, to also include the transcription itself, because the transcription itself, for example, for the transcription factors to bind to the DNA and initiate the transcription process, it's somehow something that is related to upstream of the DNA that I'm ignoring right now. So you're completely
Yeah,
right.
but don't you then also need to condition on what cell you are in? Because in one type of cell, different stuff will be transcribed and you will have different transcript abundances than in the
Exactly.
other cell type.
Yes. Exactly. You're completely right. The thing is, we our assumption here is that, okay, we have a general biology here. So I will explain the data. But the thing is, we assume that we have a general biology of the splicing. Let's say that the transcription and then the splicing that determines which of the transcripts happening here. And
Mm.
this general biology depends on the sequence. So you're right. In different cell lines and
So,
cell tissues and different cell states, we would have different combinations of protein that are going to bind to these RNAs and eventually produce these different results, right? But all of them are based on the sequence. We assume that there's a general biology embedded in the sequence. We want to extract that and then make it cell type, or let's say cell state specific.
yeah,
Did
oh,
so
okay. But I mean, we have the same DNA sequence in all of the cells, but we
Of
don't
course.
have the same RNA sequence in all of the cells.
Exactly. You're completely right. But, like, the foundation model that I trained, that I will explain, is not cell type specific.
Yeah,
It's,
and
it's
it will be a model with which we want to solve tasks like input RNA, output isoform abundances.
like, this is the one of the downstream tasks
One.
that you solve. Exactly. But the thing is, the foundation model, as I will explain, we have the RNA sequence. We want to learn proper representations for RNA sequences that contains the molecular structures. This is something novel here. What is different from a DNA foundation model that will explain, and eventually, when you perform RNA downstream tasks on the representations that is learned by an RNA foundation model, the result would be better because
No,
RNA contains some structures that DNA doesn't.
yeah. Then let's do it. Let's get to the data. We
Okay.
have 20 minutes left or so.
Okay, let's go for it. So, and I'm waiting for the transition to happen. Okay, so it was cool. Let me go back. I wanted the transition. Okay, wow.
Mm.
Okay, this is morph transition. Okay, so we have these proteins, and as I described before, here are some of the hallmarks of cancer, some of the processes inside the cell that need to be dysregulated for the cancer to grow. And these are some of them. And you can see that, like, in all of them, one of the things that is heavily perturbed is the architecture. the molecular structures that are chosen and their abundances. So I'm trying to like tell you what is transcriptome architecture and why is this important? Because if this architecture is perturbed and dysregulated, bad things will happen. This is the summary of it. So let's go for it. We want to computationally model transcriptome. And as I explained before, this transcriptome architecture is ultimately dictated by genomic sequence. And one brief recap here, we have like as we discussed with Hannes, the transcription, and after the transcription, the splicing, all of the things, the start and end site for these complex protein machineries, including the splicing machinery, to detect the start and end sites, is all depending on the non-coding, or let's say that the cis regulatory elements. Some sequence features, for example, here on the introns and exons, this splicing machinery detects it and find it, okay, where to find this intron, and let's remove it out. So everything is dictated by the genomic sequence eventually. All of these proteins, whether it's DNA bonding, RNA bonding, all of them eventually comes to the sequence and find what to do and where to start and where to end. And so genomic sequence is important. We want to learn proper, like as you mentioned, the background is ML here. We know that we need good representations in order to perform any ML task here. And so we want to model sequence NLP communities. Thanks to them, they talk about larger scale pre-trained foundation models, self-supervised learning. And then we have in genomics, we have foundation model like nucleotide transformer based on BERT architecture, and Hyena DNA based on hyenas. They try to train, like provide proper representations for DNA sequences. So the way that they work is that you provide DNA sequence, and from these models, you would get embeddings or likelihoods. And then you would use these embeddings or likelihoods for downstream tasks. And if these embeddings contains enough information about the important structures of DNA or RNA, they could be able to perform good in the downstream tasks. Okay, so however, this is where my project comes in. Model for joint representation of first full long context genomic sequence. This is something that we're trying to expand our resolution. Second, associated RNA molecular structure, which means that the exonic and intronic architecture. And second, adenucleotide resolution is lacking. So this is where my model comes in. So this is my project. RNA foundation model. We want to build or train an RNA foundation model to represent first the full long context genomic sequence, and second, to represent the associated RNA molecular structure at nucleotide resolution. In order to do that, we need two things. A training data, we need to gather a comprehensive data set of all of the possible RNA molecular structures across like human or any other species that we are interested in. So we need training data, and second, we need model that can handle long sequences and can capture the complexity of RNA molecules. Okay, so let me briefly talk about training data. So
let's rather talk a bit longer about the data and a bit shorter about the model.
Okay, great. So the thing is the model, I will skip through it, and then I will talk about the results and why biologically relevant the results are. So why we would need long RNA-seq? So as I described, we have this gene, we have different isoforms that could be transcribed. Yeah, that could be spliced from this gene. And traditionally, from let's say that all of the data that exists up until like 3 or 4 years ago, like most of the data that exists are short RNA-seq, mainly Illumina. And you can see that these short reads the resolution is an exon and flanking region. So you can see that when you have this read, you cannot precisely talk about the isoform structures that exist within a cell, not only the structure, but also the abundances. You cannot exactly talk about which of these isoform exists in a cell. You can estimate. All of the algorithms and tools here are about estimation the isoform, estimation the abundances. And you can see why here, because here a read that talks about an exon here can also talks about this exon here. So you can use like the statistical frameworks to distribute these reads. But using PacBio long reads here, each read actually is a full length isoform. So each of this read will give you a precise like description of the architecture of this isoform. And not only the structure, you have the precise abundance. You know that each of the reads is actually a transcript isoform. So this is why we switched to long read, and we generated this great, we call it C2L2 cancer cell lung long read compendium dataset, 26 cell lines from 9 tissues of origin. And you can see that the read length is much longer than short reads. The short reads is like average, let's say 100 base pair. Here we're talking about the median is around 3K. But on the other hand, you can see that the library size is much smaller than short reads. In short reads, you have like average like 50 million reads, and here we're talking about 2 or 3 million reads. So it's a trade-off between the length and the depths. And because of that, because we want to have the good of both short reads and long reads, we actually, for all of this, we actually have the cDNA match short read sequence. So for all of the cell line, we have the long reads PacBio, and we have the Illumina short reads and cDNA matched. So from the same cDNA, both of them are sequenced. And you can see that we infer the transcriptome, and you can see that we have like nearly 30% of the transcripts are novel, are not annotated, because we're working with cancer cell lines, and these are novel. So this was important for us. We want to also see different novel structures, and we want our model to also see all of this different combination of molecular structures. So have a comprehensive view of RNA molecular structures. So this is our data, and we didn't we had that data we generated. We also asked our collaborators. We have another comment. Hannes, would you please tell me if the chat is?
Yeah, I'm monitoring the chat. No worries.
Oh, okay. Okay. So we asked our collaborators to give us all of the longest data that they have. We wanted to make it as comparison as possible. And of course, all of this data are filtered for high quality reads. And so we got 149 new human samples and 46 mouse samples. And so eventually these are our training set, like not training set, I will explain. We have lots of filtrations here, but this is all of the data that we have. 2 species, nearly 250 samples, nearly 100 million long read samples, and more than 500,000 inferred transcript. When I say inferred transcript, means that different combinations of our exons and introns, different combinations of different RNA molecules structures. So this is our training set. Now let's go for the model. I'll make it fast and talking about the different like details of the model. So LoRNASH, the first RNA foundation model specifically designed to train and learn transcriptome structure using long context sequences. Okay, so let me go for it. We call it RNA foundation model because we actually working on premature mRNA isoforms. And we were focusing on transcriptome architecture at the beginning, the molecular structure, using the tokenization that we have. So we're separating the DNA content from the exonic, from the RNA content, and also within the RNA content, we're separating the exonic regions and the intronic regions. And also we're introducing like special tokens for defining the species, and also the transcription start site and the polyadenylation side, which is somehow close to the transcription termination side, because we cannot like precisely determine the transcription termination side, but we can say that okay, this is where the polyadenylation will happen, which is the concatenation of a like poly A tail that is necessary for doing the translation. So this is how we encode RNA molecular structure. So this is our input sequence to our model, and we call it long context because our input could be up to 64K. This is the version that we are working on. for sure in next versions we would increase this, and when we filter our genes and transcript onto 64K, this is eventually this is our training set. The number of transcripts are 350K on I guess like nearly 350K, and the number of tokens is 7 billion. So each token here is a nucleotide. We are talking about nucleotide resolution. And the learn, you all know this is like causal language modeling, next token prediction. So we give like 2 first tokens, ask the model to predict this third one, the fourth one, so on and so forth. So we assume that for the model to predict the next token, the model needs to capture the important structures, molecular structures within this sequence in order to be able to predict the next token. And the model is actually a StripedHyena model, constitutes of attention and hyena layers, and you all know that hyena layers are more computationally effective. They are subquadratic in terms of input lengths. And the way that it trains, you all know that like next token prediction is actually in classification problem among the available tokens, and the loss is across entropy is the negative log likelihood of the correct token given the previous ones. I created these slides for people with biological background, and the metric that they use is perplexity 2 to the power of cross entropy, and it somehow measures the uncertainty, how uncertain the model is in predicting the next token, and the smaller the better, the lesser the better. And so we perform this extensive analysis to like see the scaling laws of our model, and also to find like to find the hyperparameters that actually works for our task here. So this is like we're trying to find the optimal balance between the model size and the computational resources. And when they come model size is somehow talking about the model capacity. The bigger the model, the more complex patterns the model can handle. And here we're talking about the log E of the number of parameters. And here is the evaluation perplexity. And given a computational resources, fixed computational resources, we're talking about floating point operations here, you can see that as the model getting larger, the performance get better and better and better up until some point, and then it starts to deteriorate. And we perform this extensive task, different model, different hyperparameters, different architectures, different computational resources to see which with the given computational resources that we have access to, what is the best hyperparameters. And here you can see that thank you. StripedHyena, 16 layers, 13 layer Hyena, 3 attention, 256 latent dimension, and 7 million parameters in total. So this is our model, and this is the performance. You can see that this is LoRNASH, and this is the loss, and eventually like perplexity. And we also use the same architecture, the 16 layers, but use all hyenas, all attentions. And with the given computational resources, you can see that StripedHyena actually outperforms the all hyena and all attention, which is pretty cool. And now let's talk about, okay, we have this foundation model. The task that it was trained on is next token prediction. It's pretty cool. Now let's see that this causal language model provides 2 things for us. Likelihoods, because it's trained causally, we can calculate actual likelihoods of the sequences, and also embeddings. Now we want to investigate likelihoods and embeddings to see that whether these likelihoods and embeddings are biologically relevant or not. Is the model actually capture the information that we already know about? If the model already captured that, then we can promise the predictions and go for experimental validation for it. So here is an example. We always start with an example, and if we see it's promising, then we do it systematically and extensively. So this is an example that published in like experimentally validated and published in a Nature paper. There's a gene like green 1 in mouse, and this gene has 8 isoforms. 4 of them exclude this. This is a cassette exon situation. 4 of them exclude, and 4 of them include this exon. And this paper performed extensive like manipulation, like lots of analysis to find the splice sites and the motifs of the RNA binding protein that is actually important for inclusion of this particular exon. And what we've done is to perform mutagenesis, like performing each of this nucleotide, perform all of the possible mutations, and then capture capture, like perform a softmax to have a probability distribution of each of the nucleotides, or let's say tokens. And you can see that, and this is average over all of the transcript, that included and that excluded. You can see that the transcripts that included that exon actually mutating this splice site and mutating this RNA binding protein, RBFOX1 motifs upstream, have much more like critical effects on models' likelihood compared to the exons, compared to the transcript that excludes this exon. So you can see that the model is much more sensitive when the exon is included in the sequence. The model is much more sensitive to this splice site signals and to these motifs. So the model says that if the exon is included, mutating here is much more worse than if the exon is not included. And this is an example promising. Now let's perform systematically. So
Okay.
May I just ask the question, when you say that mutation sensitivity, you mean perplexity, or you mean that you trace like causal effect of a mutation or something further?
Okay, so okay, so as Hannes, do you have any?
Just
okay.
continue.
Yeah, we're talking about sequence likelihoods here. Okay, so let me see. I have
I see. So
slide.
this is perplexity. This is just
This is
probability of
sequence
docking.
Yeah, let me show you this. Yeah, this is sequence likelihood. So here's a slide for it. So when we have a sequence here, we can actually calculate the likelihood of each of these token. And because this is the likelihood, the likelihood of the entire isoform sequence is the sum of the likelihood of each of these tokens. Okay? And I'm talking about the likelihood of the sequence. Here, when I show you this, when I show you, if I can find it, yes. When I show you this, I mean the likelihood of the sequence after the mutation is not the likelihood of the token. I perform the mutation and then calculate the likelihood of the entire sequence. And I do this for all of the possible mutations. And then I have 8, because the possible tokens here are 8, I have 8 sequence likelihood. And then I perform a softmax, convert this into a probability distribution. And here it says that, okay, we have 4 transcripts. For each of them, we have 8 possible mutations. So here it says that the likelihood of this token is almost 1. Means that if you mutate this token in the transcript that include this, the likelihood of the entire sequence will drop significantly. Does it make sense, Dennis?
I see. Yes, it does. But you still need to track only the likelihood of the subsequent tokens. So if you mutate at the start of the sequence, then it will naturally affect the whole sequence more, it seems. Or maybe
exactly. That's the thing. There are some other causal models that what they've done is to just look at the input the like entire sequence, and they just look at the, as I explained here, they will just look at the likelihood of this of the correct token. But the thing that we've done here is to perform the mutation and let this effect propagate throughout all of the sequence, and then calculate the whole isoform sequence, whole isoform likelihood.
Okay, thank
you. Okay, so let me make this full screen again. Okay, so now let's make this systematic. So first, we're talking about splice sites. So here we got, this was just an example, but here we got all of the cassette exon situation in our transcriptome and perform the mutations on the splice sites on the transcript that include that exon and the transcript that exclude that exon. And here we're talking about accept and donor. These are like 5' and 3' depending on the strand, but it's just like, let's say that the start side and end side of this introns that are flanking the cassette exon. And you can see that the delta likelihood when we perform the mutation. So here we're talking about the delta likelihood. I have a reference sequence, which is the reference isoform. I perform the mutation, on the splice side, and I have another sequence. I calculate the likelihood, and I calculate the delta likelihood of the reference and the mutant. You can see that the transcript that includes the exon are much more sensitive to changes in the splice sites versus the transcript that exclude the exon. And this shows that systematically that our model actually learned about the splice sites. Pretty cool. Now let's go for the other part. So here you can see that this like RNA binding protein RBFOX1 that I showed that the model actually paid attention to the motifs. Here we perform a systematic analysis. We again got all of the cassette exon situation, and we have this flanking areas. And I got the RNA binding proteins regions from ENCODE, all of the RNA binding proteins, and find the overlaps between this region. For this analysis, we exclude the splice sites and the 5' nucleotides like upstream and downstream. We don't want to do like find confounding factor from the previous analysis. So this is talking about only the RNA binding proteins. And we found all of these overlaps and just deleted. And by deleting, I mean replacing it with N, with the unknown token. And this is the control positive, how different RNA binding proteins will affect the likelihood. And then as a negative control, for each of these regions, we find a random region, but it doesn't have any overlap with the RNA binding proteins. Random region, we then delete it, replace it with N, and have the negative controls. And here you can see that for all of the RNA binding proteins, and the first one is the random ones, we have box plots. We have box plots of delta-like likelihoods. Why box plot? Because each RNA binding protein would have many overlapping regions, binding regions for cassette exons. And then what we've done here is pretty cool. We sorted all of the delta likelihoods based on their median, and then look at the enrichment of the splice genes that we call it splice factors, the RNA binding proteins that we know that are important in the splicing machinery. And as you can see, as the delta likelihood increases, the enrichment in the splicing factors also increases, which means that the model somehow understood that these motifs are important for the splicing of these cassette exon. And this is pretty cool. Like this is systematically shows that the model understood about the nearby, or let's say the RNA binding protein in the intronic regions, flanking intronic regions that are important for cassette exon inclusion or exclusion. Pretty cool. Next analysis that we've done. Sorry. Okay, then.
Oh, wait. So
Yeah.
Ali, wait a second. So these two experiments now have been both like let's prod at our model and see what does it capture these things about RNA already, like just the vanilla trained model. But then if we maybe let's go away from like let's believe that your model captures something about RNA, and we've seen evidence for these two things now. Then do you have any experiment or so where you like say this is the most useful thing that my model can do, or the most like a new discovery, like a new bio discovery or something like that? Or what would you say is if not, then what would you say is the most impressive thing that your model captures?
Exactly. Okay, so I guess I will talk about both of them in the next slides. But the thing is, all we're trying to say here is that we have a foundation model, and the likelihoods and the embeddings, the representation that this model is providing contains the already known biological knowledge. This is proof that the representation is good. And then we took these representations and likelihoods and try to perform downstream tasks. And these downstream tasks are, as you mentioned, one of them is the transcript abundance prediction. And yeah, I guess like the most impressive part is that I guess the most impressive part is the sequence generation, the last part. I'm pretty curious to see that. And we have lots of things to experimentally validate, but currently, like this manuscript that we're writing is the data and the model. And then in the next, we're going to experimentally validate the novel things that we found. For
Okay,
example,
then
uh, yeah,
if you already have some novel things or so, then let's maybe like because I need to leave in like a minute or
okay,
two. Um,
okay, sure.
let's
Sure.
maybe the one result that you want to highlight.
So exactly like the next result is something that like here we had this previous experiment, but instead of RNA binding proteins, we have a window of 20 base pair and just with a stride of 10, and we're just mutating, like deleting regions, different regions nearby the cassette exon situations, and then find the delta likelihood, and then take the regions that have the highest delta likelihood, and then perform the de novo motif discovery. We found 8 motifs that our model is sensitive to it. 3 of them are known. These are the 3 of them, and 5 of them are not known. And whether these 5 new motifs are biologically relevant, or which RNA binding protein are binding to this, or any other, we don't know right now. But from these de novo motif discovery, 3 of them are already known. These are the 3 of them, and these are 3 already known as splicing factors. But 5 of them are novel, maybe biologically relevant, maybe not. But we will put them. I can show you the other motifs if you're curious to see. We will put them in the supplementary. But
Okay,
so
so you discovered
these are
motifs, you discovered binding sites.
like binding site. These are motifs that when I delete them, the model gets angry. The model says that the likelihood drops a lot. And these 8 motifs, 3 of them, we already know that these are known binding proteins, but like RNA binding proteins binding sites. Okay,
Yeah,
but 5 of them are not. Whether these are biologically relevant or not, we need to like
yeah,
do mutations and see if the expressions happens or not.
cool.
Okay, okay.
Then Ali, I would do it the following way that like from my side, a big thank you and sorry that I have to leave early, but for me this has been super helpful and I also just love your energy in presenting and chatting with each other. So maybe we can have an offline like online offline chat at some or maybe offline chat if you come to MOML or some conference around here and at MIT. But we should do that. But then I would leave you guys alone now. And maybe Denise, are you still around? And you had some questions. Do you maybe want to ask them? And then once you guys are done, you can just call the day.
Yeah, I think maybe if the presenter wants to finish the presentation, everyone would be
Exactly.
happy to listen.
Yeah. So then maybe Ali, you can finish the recording. We'll keep it going, and then
Mhm.
I'll just at the yeah
Of
we'll
course.
just keep the recording going and I can watch it also later and we'll put the whole thing on YouTube and you can answer some questions of Denise and others afterward.
More than happy to. For sure. Have to see you, Ernest.
Perfect.
Take
Then
care.
see you guys around. Bye.
Take care. Goodbye. Okay, so let me continue. And then, so all of this previous slide, I try to convince you that, okay, our model is cool, the embeddings, the likelihoods are cool. And then now we want to perform some like tasks, let's say 0-shot prediction task at the beginning. And why 0-shot? Like, again, we're trying to convince people that C, the likelihoods are informative. C, the likelihoods contains biological information. So this is a good foundation model. So the representations that the model provides is cool and it's good. So 0-shot is one of the ways to show that. And here we focus on likelihoods and 2 types of likelihoods, whether the likelihood of the P token, the pass token, the poly A tail token. And the reason for that is that it's actually for the model to predict this token, the model needs to see all of this previous tokens. So this is somehow like convention in the causal language modeling that they usually take the last token as the representative token for the entire sequence. Here, we took the last RNA token of this isoform, as we call it the likelihood of the P token, or the likelihood of the entire isoform, which is the sum of the likelihood of all of the token. Okay, so this is two representative of isoform sequences. So I will go fast through these tasks, and feel free, because it's hard for me to read the comments. Would you please unmute yourself and just interrupt me? This would be easier for me. Thank you very much. So zero shot, the first one, predicting absolute abundance. There's no fine tuning or training models. This is the experiment. We have different isoforms. Each gene would have different isoform, different sequences for that isoforms. And based on the C2L2 data set that we have different expression and abundances in different tissues and cell lines. We took the average. So we have the abundances, the average abundances of all of these isoforms. On the other hand, we use LoRNA to get the likelihood for each of these isoforms, get the likelihood of the P token. And then we calculate the correlation between these likelihoods and the average abundances. And these are the results of LoRNA and the two other DNA models. We're trying to explain that when LoRNA sees the different architecture, the RNA molecular architecture, it has extra information compared to the DNA foundation models. And here is the correlation. All transcript is almost 0.15. Again, this is zero shot. And here's within genes. So for each gene, we have different isoforms and different abundances. We calculate a correlation for each gene. And we have nearly 20,000 genes. And this is a box plots of all of those genes. And these are like our models within gene correlation are significantly greater than the DNA models. So this is pretty cool, given that it's zero shot. And another zero shot task that we perform is a trap exons abundance and counts. And trap exon is as an experiment. It was hard for me to understand it. First, it's basic biology, but for me, it was difficult to understand at first. So we have a vector sequence. It's predefined, pre-engineered, and they will like split the DNA sequence randomly, and they put these split regions, like random regions, within some particular regions of this vector sequence. And then when they inject all of this into these vector sequences, they put these vector sequences in different cell lines. And then they will see that which of these regions, random DNA regions that are included, which of these regions would end up inside the final product. And they call it, this region is trapped as exon. And then they calculate they measure the abundance, so the count of this trapped exon. And the way that we formulate this, because our model actually can distinguish between the exon and introns, we got this we have this vector sequence. And when we want to inject this random fragment, one time we inject it with the region trapped as exon based on the tokenization. And the other time is that not trapped as exon, it's an intron. And then we have the sequence likelihood of these two sequences. And then we calculate the delta likelihood. And then we find the correlation between the delta likelihood and the abundances. And this is the correlation. So, and the data, we got this data from this cool research of U of T, that they performed this huge experiment, and they found like more than 1 million autonomous, like trapped exon region, randomly trapped exon regions with different abundances. And this shows, again, the previous experiment and this experiment shows that the model somehow have some clues about the chance of existence of one particular sequence in the nature solely based on the sequence structure that the model trained on. So because these are all zero shot. So again, pretty cool, zero shot. And the final zero shot prediction task that we perform is the ClinVar non-coding SNVs clinical significance predictions. And the way we've done that is something like this. For an isoform, so this is an isoform, and this is the sequence, and this is the reference sequence. We have the likelihood based on the LoRNA. And we perform the mutation, and we have the mutant version, and we have, again, the sequence likelihood of the entire sequence. And we have the delta likelihood of the reference and the mutant. So this delta likelihood of this isoform in the reference versus mutant version is actually the score that we're giving to this particular mutation, okay? So we have a data set, ClinVar, non-coding SNVs. We particularly focus on non-codings because for the codings, we have many tools that are actually working well. And for the coding to focus, like when you provide intronic information for the model, you're somehow distracting the model from the coding regions. Because for the coding regions, in order to like for the coding regions, it's much more easier because you have the codons and you can estimate the translated protein, and it's much easier task. But for the non-coding SNVs, we have this ClinVar data set, and we have 5 labels: pathogenic, likely pathogenic, VUS, benign, likely benign, and we actually categorize them into 2 labels, pathogenic and non-pathogenic, and we perform this binary classification. So for each of these mutations, we have 2 labels, pathogenic or non-pathogenic, and we have this score, this delta likelihood, and like without any training, any model, just 0 shot, we're trying to classify based on this score. This is the result that we put on manuscript, and we reevaluated and realized that it's wrong. And this is the updated result. And for now, I just have Splice AI and LoRNA because these 2 models are great. The DNA models, Hyena DNA and nucleotide transformer, are not similar to these. And you can see that Splice AI actually outperforms us here. You can see that the AUROC and the AUPRC is better, slightly better than our model. But when we exclude the splice site mutation, so pay attention to this, that the Splice AI is actually trained on this task, on the task of predicting the splice sites. So the model knows a lot about the splice site and the flanking region. Here, we remove the mutation, the non-coding mutation that are closed, that are on the splice site or within 5 nucleotide window of the splice sites, and then perform this. And you can see that our model likelihood 0 shot is actually outperforming the Splice AI, just by like a small margin. But again, this Splice AI model trained on this task, the Splice AI prediction, but our model just 0 shot based on likelihoods. And this is pretty cool. This shows that our model actually knows about non-coding regulatory elements. And when the mutation happens in those elements, the model got sensitive about it. Okay. So now let me talk. Okay. So I guess, let me see the time. Okay. It's almost 1 hour. I tried to make it fast. So the previous slides were all about likelihoods. We're trying to convince people that our likelihoods are cool and informative. Now let's talk about embeddings. Usually on causal language modelings, people work with likelihoods, for example, the EVA model or Hyena model. But here we also wanted to check the embeddings. And here the analysis that we perform, when we have a sequence, we have 16 layers, right? And each of these tokens have embeddings in different layers. And so the question is that which embedding, which layer, and how you want to generate an embedding for the entire isoform. Therefore, people get the average embedding in 1 layer or different layers. But for now, here we don't want to find the best embedding possible. We just want to show that these embedding contain information. And we got the P tokens embedding of the 1 to the final layer. So the final layer is specialized in next token prediction. Like the 15th layer, we got the past token, the P token embedding of the 15th layer of our model for all of the isoforms, and perform this task that I will talk about. Here you can see that the dimensionality reduction, the UMAP representation of this token, and then we cluster them, and there are different clusters. And when we do the motif discovery, we realize that the embeddings actually contains information about different motifs in different clusters. So again, this embedding contains information about regulatory elements, which is pretty cool. So here, what we've done, this is not 0 shot anymore. We're trained model, downstream, but not really complex model, not neural net. Here, what we've done is that, okay, so for each isoform, we have an embedding, right? And here we have 26 cell lines. I trained 26 models, gradient boosting regression models. For each of these models, the model input is the isoform embedding, and the output is the abundance, the absolute log TPM abundance inside this particular cell line. So it's a specialized cell line specific. And you can see that this is the Pearson correlation, bar plots of Pearson correlation of the observed absolute abundance, log TPM abundance versus the predicted ones. And this is the best one, the SW480. the colon cell line, and you can see that it's pretty cool. So we're taking the embeddings and we're doing the predictions, and we're getting good results. And this is all promising. Again, we can investigate a lot on how to generate embeddings for this isoform, but just getting that embedding, we're getting this result. And another analysis that we've done.
Sorry, can I clarify? So the abundances that you show here are for cells. So for not cells but tissue types. Or
Yeah,
you mean that they have a particular isoform inside the tissue type? Because I guess for the second one, it's hard to predict from the same embedding.
Yeah. I mean, here I just colored them based on different tissues, but each of the model is based on the cell line. So this is a model talking about the abundances in this cell line. This is another model. This is another
I see.
So the input model is similar. All of them have the same embeddings, but the output, the target variable, is the absolute abundance in that particular cell line. Did I answer the question?
Of a cell line or of a transcript of a cell line, right? So essentially, the model tries to predict. It looks at a whole bunch of transcripts and it tries to output the abundance of a particular cell line.
Can you ask me again? Sorry, can you ask me again?
It looks at a whole bunch of transcripts at once because it's gradient boosting, or it looks at a single transcript, at a single one.
Single, yes. The input is the embedding of a single transcript, and the target value is the absolute abundance in that particular cell line. Right? This is input output. So the training samples would be like here I have 1,000, for example, in this transcript, I have like in this cell line, I have 140,000 transcript expressed, right? So when I have the training set, I like the split is like this. All of the chromosome except the chromosome 10 and 8 is in the training set. Chromosome 10 is the validation and chromosome 8 is the test set. These are the result on the test set. I train the model. Each sample is that the model get an embedding, try to predict the absolute abundance of that isoform, and then the next isoform, and then the next isoform.
I see.
Each sample,
Yeah, this is interesting. So it means that it learns from embedding some representation that essentially is uniform for like universal for different isoforms that are enriched in a particular cell line, and it learns how to somehow to extract those representations, and based on them, on a holdout set of transcripts, it manages to correctly predict the abundance. Yeah, it's interesting. Okay, thank you.
Yeah, no worries. And the thing is, again, I want to emphasize this is just to show that the embeddings contain information. Again, we can play like another project how to find a good embeddings from this. And this is, again, we could also fine tune our model, like the neural net, the foundation model that we have. This is not fine tuning. This is frozen embeddings. We got it, and then trying a gradient boosting regressor on top of it. So we can make this much better. Actually, like right now, there's another PhD student that working on this project, specifically this part of the project, to make it better. How we can make this relative abundance, absolute abundance prediction better. Okay, so another task that we perform on the embedding to show that these embeddings can also be used in comparing cell lines. So within each tissue, we have 9 cell lines, we have 9 tissues, and within each tissue, we have different cell lines. And this is here I show only two tissues, but we have result for other tissues too, and we will put that in the supplementary. So the thing is, here I'm trying to what is the prediction task here? Here, the prediction task is a binary classification. So let's say here I have it like comparing this HCC44 comparing with H358. When I'm comparing this cell line versus this cell line, I have a set of isoforms. Some of them are significantly upregulated in this cell line. Some of the other isoforms are significantly upregulated, differentially expressed by higher expressed, upregulated in this cell line, and the rest like no changes, no significantly changes. Okay, so here when I'm comparing this cell line versus this cell line, it's a binary classification. the model will take the embedding of the isoform and tries to predict whether it's upregulated or not. So you can see that it's not symmetric. This one, this cell line versus this cell line tries to, the true ones, the label 1 in the binary classification, we have 0s and 1. The ones are the isoforms that are upregulated in this cell line. When I'm comparing this one with that one, with HCC44, it's actually the ones, the true labels, the positive samples are the isoforms that are upregulated in H358. And this experiment tries to show that this embeddings contains information enough that can distinguish differences between cell lines. Again, here is a random forest binary classifier, and we can perform fine-tuning on the entire foundation model to get much better results. But based on this pass token embeddings, you can see that we're actually doing pretty good in this binary classification. Okay. So, and the final task, sequence generation, which I think this could show lots of potential with this could be pretty cool, is that this model trained causally, so we can generate sequences. And the way it works is that we provide a prompt, for example, H and S, so that we're telling the model generate human sequences and S, and start from the transcription start site, and the model starts to generate. And as you all know, is that, let me make it laser, it's cooler. So the model generates next token, and then we provide all of the sequences again to the model, next token, up until the model generates the P token, the pass token, and then it's finished. And then we have the entire sequence. And then we try to compare the generated sequences with the references sequences, the human references sequences that we have in the reference annotation. And the exon length, intron length, number of exons, and sequence lengths are highly similar, and it's pretty promising. I can say it like zero-shot sequence generation, because when you want to generate sequences, lots of parameters are here: temperature, top P, top K, lots of like parameters you can, hyperparameters that you can train and you can tune to have better and more meaningful results. This is just getting the default sequence generation parameters, which is a temperature 1, top P of 0.90, I guess, something like that, and then generate the sequences. And here, the final one, the splice AI probabilities. So a splice AI will give probabilities for each splice site. And here, we apply a splice AI on reference transcript, and you can see that the distribution and on our generated sequences, and then we perform like these two distributions are not significantly different, statistically speaking. And this is pretty promising that our model actually generates sequences that another tool says that these are legit and correct, high probable splice sites. And one other like analysis that we found is that, okay, we generate like here for this, we generate 12,012, this random number, 12,012 sequences here. And after we have sequences, okay, and our sequences contains like exonic and intronic regions, and we do the splicing. How? We remove the introns, then we have the mature mRNA, and then we did the ORF finding, and we have the ORF. When you have the ORF, you can predict what would be the translated proteins. And then this is for the reference human transcript codons usage and the generated codon usage, because codons are not randomly distributed. We have 64 codons. It's not uniformly distributed. The usage this is the distribution of usage of different codons. And our generated sequences, the codon usage, the correlation is 0.63, and this is pretty cool, because the model, like if you want to train a model to understand about the codons, which we have models like I guess Codon BERT is out there, like you provide the codons to the model and the model will learn about the distribution. But here we're providing raw pre-mRNA to the model, and exonic regions are a small portion of the pre-mRNA because we have long introns, and still the model understood about the different combinations of this amino acid and the correlation. This is actually was really really surprising for us. We didn't expect 0.63 correlation, but this is cool, and this is why I'm telling that this sequence generation It could be super cool that we generate de novo sequences with like consider that we train our model to generate sequences transcript with particular functions, and this would be super cool. And
It's okay. If you want to finish, I can postpone the question. But honestly, the question here is simple. You mentioned that you train on both human and mouse data. Here, the model is also trained on both, but you are only using the reference for human.
yes, I only have generated human sequences. We can do this for mouse too, but because of the sequences that we generate, the prompt was always H and S. The sequences that the model generated supposedly was for the
I see. I missed this prompt. Yeah, I see. Makes sense.
Okay. Yeah, great. And that's it. Like this is LoRNA SH, LoRNA long RNA and SH for StripedHyena. We might update the model with different architectures in the future. And it's pretty cool. I try to convince you all that the likelihoods and the embeddings are pretty cool. This if you want to use if you want to have likelihoods and embedding for any RNA task, come and use our model. And this is the previous version of our manuscript published in bioRxiv, but by October 21st, we will publish the final version with all of this news results and also some other new analysis. And we will also submit it to journals, and we also put our models, code, scripts, ways, everything online and open source, and you can use it. And I want to acknowledge my supervisors and all of the collaborators and everyone that helped me. And thank you so much. These are two cool lab members, cool labs and cool lab members. And thank you so much. And I'm here for any other questions that you might have.
Yeah, I would just like to add, it's amazing work, actually. I actually just want to say that I could also see potential in bioproteins, actually trying to find the genes for certain bioproteins, since you are generating potential introns and extrons. So you can highlight those in the species genome, and then from there, go ahead and do the actual testing to see if that particular region is, in fact, a genome. So yeah, that seemed very promising.
Great, thank you. Makes sense. Thank you for the comment.
Yes, and I have a couple of simple questions, I guess. The first one is, have you considered actually using as an additional data some class label on the either like disease modality, like phenotypic modality, or cell type modality, so that you can essentially easier steer the generation of transcripts in a way that it will correspond to, for example, a particular cancer in a particular cell.
Of course. Like the other model, like we use this StripedHyena because of EVO model. I don't know if you're aware of EVO. It's based on the DNA bacteria. And it's a huge model, like 7 billion parameters and trained on 300 billion tokens. But the thing is, because they have different types of bacteria, they also have tokens for these bacteria. So when they want to generate, they will in the prompt they will provide the type of bacteria, and they will condition the model to generate sequences for that. This is actually like completely right, and is a great idea to like have human and then talking about this particular cell line, this particular tissue, or whatever, this particular cell state. And you're completely right.
Yeah, I think this usage of essentially control tokens to pose a generation is very natural, and if you have enough data, it can work. But the last question is again about data. So you mentioned that you have about 300,000 transcripts. You mean by those transcripts that, so essentially, if you have the same isoform with the same composition of exons, but a single mutation, do you consider those two variants different transcripts? Like if you have a single nucleotide mutation?
Yes, actually,
yes,
like theoretically speaking, yes. But the thing is, here we're talking about RNA-seq. long read RNA sequencing. When I'm saying a long read RNA sequencing, it's you have this reads and you would map it to the genome, okay? And the thing is, if the mutation end up like we have another analysis that we will include it in the I can show you now, or no, I don't have the figures now. So the thing is, if that mutation will lead to changes in exonic and intronic architecture, we would have it as two different isoforms. But if it's not, no, we would have it at one isoform.
I see. And you consider it a single transcript, but in two variations then, or not? Well, that was the question, not really about isoforms, but about the size of the training set, the diversity.
Yes. It's when I say 350,000 means that 350,000 different combinations of exons and introns.
I see. Then the data set is quite large. Yeah. Okay, that's great. And then the last question, I guess, is about the train test split. I guess in the paper you probably detail it, but the experiments that you show with the likelihood, they do look quite promising. But the question in biology, I think, is always about how you made the test train split, and if you even do it, because in language modeling we usually don't, kind of. But so did you report results on the test here on the whole data?
Yes. Like if anything, like for example, if anything that is trained, it's on the test data. But for example, like these are all test data, right? This is zero shot talking about no. For example, this is zero shot. Like there is no testing here. There's no training.
Yeah, I was talking about likelihoods. I was talking about this likelihood comparison.
You mean here? Let me see. Like for example, which one?
Even before, I think.
This
yes. I think this one might do. Yeah, maybe.
So for example, this one that we're talking about, there is no training. You might mean that, okay, so this is it. For the training and testing, when I was training the language model, I didn't split anything. So at the training, when the model was training, the model had access to everything except chromosome 10 and chromosome 8. Okay, that was the that I provide here information. Okay, so you can see that this is the evaluation loss, which is the test loss, which is on the chromosome 8 only. Okay, but after I got this, I trained the model on, so for example, here, you can see that there is no testing and training. I have this green one gene. Oh, so you mean that maybe this green one gene is in the chromosomes that the model trained on. I'm not sure. I haven't checked for it that whether the green one gene is on it, but this is not on the chromosome that is for the testing. This is on all of them. Okay.
Yeah, so, specifically for likelihoods, it is interesting to understand how model comprehends the global structure of a transcript. If it can, for example, detect some motif early on, and based on this motif, understand what kind of motifs can be present at the end. And then, of course, if it is done in the context of the same gene, or of maybe two very like genes from the same gene family, because there are like duplicates of genes, then it's one thing. But if those are completely two different genes, then it's much more impressive.
The thing is, I can tell you this. I'm trying to remember what we've done. Anything that is based on some training, we have training, validation, and testing, and the split is always the same. All chromosome except chromosome 8 and 10 is in training. Chromosome 10 is validation, and chromosome 8 is testing, and it's like 95-5. This is somehow convention. Anything that needs training. But anything other than that, anything that is zero shot, anything that is not related to training or validation, it's the entire data set. Okay. So here we're talking about training and validation. So all of this result is based on chromosome 8. And here, this is just one green. I can also check here. So green 1 gene is on one, which one? Which chromosome? Green 1. So no, this is not mouse. This is, so let me, if zoom, let me, green 1 gene on mouse. So how can we see? So let's go for this one and see which chromosome is on this. If I can search for chromosome, no, chromosome 2. Okay, so it's on chromosome 2. So this gene was within the training set. But here, this is,
Yeah, thank you. I got it. Okay, that's still very cool. Maybe I will check that generalization myself, because it's really interesting how much you can get with generalization. Thank you for answering the questions.
Of course, in terms of like genes, like we got it here. I was running the evaluations, and it was like 1, 1, 1, 1, like AURC on all of them were 1, 1, 1. Like the worst one was 0.99. And I realized that the split that I made was wrong. It wasn't based on chromosome. It was randomly splitting chromosome. And exactly what you said happened, like data leakage. When you distribute the isoforms of a gene within training and validation or testing set, it's absolutely data leakage because the patterns are similar. But in all of the training and testing sets, like it become too good to be true. When you don't split based on chromosome, it become too good to be true. You will understand that something is wrong.
Yes. I just think that some people try to do splits on gene families, but I don't know, honestly, how popular it is today. It was more popular, I guess, some years ago already. But thank you for answering.
Gene families are different chromosomes, like one genes within one.
I think they can be. That's the problem. But genes from the same family can be on multiple chromosomes, because they can duplicate, and then they can essentially translocate to another chromosome, and then you essentially get a kind of data leakage, because you have two very similar genes, and you learn patterns from a single gene, and you learn to associate patterns from a training set to abundance, for example. And then you have a very similar gene on another chromosome, and it kind of translates. But yeah, I don't know how much of an impact it has, to be honest. And it does depend, obviously, on the chromosome and so on.
I guess I can check this. Like, if that's the case, this is actually like data leakage in a minor level if we have similar gene families. And the thing is, I can check here. I can search for gene families, like this task, for example, okay? And I can search for gene families and see that if the prediction of a gene that has like from the same gene family that is in training and is in testing, if the predictions are better than the other genes. So we can see that if the model is using. Thank you for mentioning.
Yeah, it's interesting. Although I would say that it is not a given that two genes from the same gene family will be they have the same abundance, for example. They may, but sometimes they can be, for example, they can be very similar, but they can play an antagonistic role. Like one actually inhibits another one. Then maybe their abundances should be different. But yeah, okay, thank you. I think we can close up this, because it's already 1 hour, half an hour past. Thank you for a wonderful presentation. Really enjoyed your work and really want to investigate it further. Okay, bye. Thank you.
Nice to see you. Take care.
I don't know how to end. I guess I'm a host now. Or am I? Yes. So I will I will click end then. Bye.
Great question. Very much. Thank you very much. Amazing talk. Would you be able to share the slides maybe on our Slack or somewhere, or send it to Hannes?
I'll see. Yes, but like we're going to publish everything by October 21st. So before that, I'll check, but after 21st, you'll have access to everything.
Okay, it would be great. Thank you very much.
Thank you very much. Take care.
Okay. Thanks again. Bye. Have a nice evening.